This is part 3 of the PowerPoint slide set 2B. We are now going to learn about proteins. Here we go. So the 4 macromolecules, or the 4 large groups of organic molecules that are important in living things, are carbohydrates, lipids, proteins, and nucleic acids. We've already learned about carbohydrates and lipids. So proteins make up 20 to 30% of cell mass. In other words, 20 to 30% of the structure of our cells. Proteins can be part of our cell structure, like, just components of muscle tissue, bone tissue, that kind of thing. Proteins also function as enzymes, which speed up chemical reactions. And there are proteins that aren't just part of the muscle structure, but that cause muscle contraction. So they basically serve 2 roles. Proteins contain the elements carbon, hydrogen, oxygen, and nitrogen. And then sometimes they will contain sulfur and phosphorus. That P stands for phosphorus, not potassium. That S stands for sulfur, not sodium. So the elements you find in proteins are carbon, hydrogen, oxygen, just like in carbs and lipids. Then you're going to add nitrogen, and sometimes you will have sulfur and phosphorus. The amino acid building blocks, or monomers, are held together by peptide bonds. And proteins have a very complex structural shape. We talk about proteins in I already said peptide bonds. Okay, we talk about, here we go. We talk about proteins in 4 different levels of structure. So the first level of structure is the primary structure, which is simply just the sequence of amino acids. There are 20 different amino acids. So the sequence of amino acids that make up the chain of what we call it now a polypeptide. So the primary structure is just simply a chain of amino acids. It is the sequence of amino acids. Then you take that chain and you shape it into either a spiral or helical structure, which we call an alpha helix, or it can be pleated or folded, like if you were to make a paper fan, and that's called a beta sheet. So the secondary structure is either an alpha helix or a beta sheet. And then the tertiary structure is you take those alpha helices and beta sheets and you fold them and twist them and loop them into a 3-dimensional shape. I always think about it like this. So if we go back to the secondary structure, if you remember what a phone cord looks like, I know you guys have seen one. There are some phone cords still left, especially in offices and places like that. You may not have one in your house, but cords that are spiral that you see, not just to phones, but there are different appliances that have cords that have a spiral shape. Okay, so you take that spiral cord and you tie it in a bunch of knots, then you have the tertiary structure. So the tertiary structure is the final 3-dimensional shape of the protein. Now, some proteins are even more complex, and that is when you have 2 or more, there's 2 different ones here, 2 or more tertiary structures chemically linked together. So that is when you have the quaternary structure. So let's go back. This is showing you the different types of proteins. We have structural proteins like collagen. We have enzymes. We have transport proteins like hemoglobin. And here you can see that hemoglobin has the quaternary shape. There are 4 subunits, not the red atoms of oxygen. Ignore those. but the folded globular subunits here that are linked together. So the hemoglobin has 4 subunits that make it have the quaternary level of structure. It's got 4 tertiary structures linked together. Okay, then we have contractile proteins like actin and myosin, which the myosin will attach to the actin and pull on it to cause muscle contraction. We have proteins that function as chemical messengers, such as there are certain hormones that are proteins. Some hormones are steroids, but some hormones are proteins. Insulin is a protein hormone, and it helps to regulate blood sugar. Then we have antibodies or immunoglobulins. They're shaped like Ys, like upside down letter Ys. And antibodies will help to destroy certain pathogens like bacteria and viruses. Now, amino acids are the monomers or building blocks of proteins. There are 20 types of amino acids. And the amino acids are joined together to form the protein, and they're joined together by a bond called a peptide bond. So what they're called when they're linked together in a chain is a polypeptide. Now, an amino acid looks like this. It has a central carbon, then it's got a hydrogen group, an amine group, which is a nitrogen and 2 hydrogens, and then an acid group, which is a carbon attached to an OH and an O. This is called a carboxylic acid group. And then you've got any chemical can go here. Any chemical or group of chemicals can go here in place of the R. So the R group is what makes the amino acid. There are 20 different R groups for the 20 different amino acids. So all of those different amino acids are joined together by a dehydration synthesis reaction. A water is removed from the 2 amino acids, and they're joined to form a dipeptide, and water is given off as a product. And the bond that joins them is called a peptide bond. So for the levels of structure, we have the primary level of structure, which is just the sequence of amino acids. The secondary level is going to be a helix or a pleated sheet. Tertiary is the 3-dimensional structure of the helix or pleated sheet, or it can be a combination of both. And quaternary is when you have 2 or more tertiary structures joined together. All proteins don't have the quaternary level. The animation about proteins is found on slide 56. I encourage you to watch that. The way that enzymes work, this is an example of, it's a graph that shows the energy that's required to convert reactants into products. Okay, so you start out with 2 reactants. Without an enzyme, it takes a certain amount of energy for those reactants to react and then form into products. So this is called the activation energy. Now, with an enzyme, that activation energy is lowered. So if less energy is required, then it takes less time. So the way that enzymes speed up chemical reactions is they lower the activation energy, and that causes the reaction to happen faster. Here you can see an illustration of an enzyme. It's got 2 areas on it called active sites. The substrates, in this case, they're amino acids. The substrates are the reactants. The substrates will combine with the enzyme. The enzyme will facilitate the joining of the 2 amino acids, so it will cause the chemical reaction. And it will speed up the process. It will speed up the chemical reaction by lowering the activation energy that's required for those amino acids to combine. they're now going to form into a peptide, and then that enzyme can now be reused. It can be used over and over again to cause the same reaction with 2 different amino acids. Now we're going to move on to our last category of organic macromolecules, nucleic acids. And nucleic acids can be DNA and they can be RNA. DNA is our genetic material, contains the blueprint for our traits. Nucleic acids can contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. So these are the elements you'll find in nucleic acids. Nucleic acids are the polymers. The building blocks or monomers are called nucleotides. So there are billions of nucleotides in a DNA molecule. A nucleotide consists of a base, a nitrogen base, we just call it a base is fine, a sugar, which is a pentose sugar, and then those H's again, it should just be little h's, but a phosphate group. Okay, and the 2 classes of nucleic acids are DNA and RNA. RNA is ribonucleic acid, DNA is deoxyribonucleic acid. DNA is forms a double helix shape. That means it is a twisted ladder. It contains a deoxyribose sugar, phosphate group, and one of 4 bases. The bases can be adenine or guanine. They are called purines because they are a double ring shape. They have 2 rings in their structure. Or the bases can be cytosine and thymine, which are pyrimidines because they have a single ring structure. And since you have 2 strands of DNA, one strand has bases that are complementary to the other strand. If you have an A in one strand, called the template strand, then the complementary strand will have a T. If you have a G in the template strand, the complementary strand will have a C. This is called complementary base pairing, and that allows us to know the sequence of the DNA. If we know the sequence of the template strand, that allows us to know the sequence of the complementary strand. So A pairs with T, G pairs with C, and you can see that here. Here is our twisted ladder shape, and you can see it broken down into, up here at the top, 2 nucleotides paired together. So the nucleotide on the left contains the base adenine. So it's got adenine and then the deoxyribose sugar and the phosphate. So the one on the right that pairs with it is going to be thymine. And that thymine, you see how it has a single ring structure? That makes it, that's because it is a pyrimidine. And then the adenine has a double ring structure because it is a purine. And then the nucleotide that contains thymine contains also a sugar and a phosphate. So the sugars and the phosphates form the sides of the ladder, and then the bases form the steps or the rungs of the ladder. And the ladder is twisted, and that's where you get the double helix shape from. RNA is just a single strand. It doesn't form a helix. And there's an animation on DNA and RNA I recommend that you watch. And that is slide number 73. Then we have a little bit of information on ATP. ATP is actually composed of adenosine. Okay, so ATP stands for adenosine triphosphate. So it contains the nucleotide adenine. It's like a modified adenine. And ATP contains adenine and 3 phosphate groups. So here's what it would look like. It would be adenine attached to a ribose sugar, attached to 1, 2, 3 phosphate groups. That's what makes it triphosphate. So when one of these phosphates groups the bond is broken and it's released, that's when you get energy from ATP. ATP is the molecule that our cells use for energy, and our cells can get that energy as soon as the bond between one of those phosphate groups is broken. So when ATP is broken down into ADP, that's adenosine diphosphate, and gives off an inorganic phosphate group. One of those phosphate groups is given off. That's what this means here in the purple circle. When it releases a phosphate, it also releases energy. So basically, when ATP is broken down, we get energy from that. And that energy can be used to transport molecules across the plasma membrane, for muscle contractions, for the work of certain chemical reactions that require energy. And that will be it for chapter 2. This is the end of the chapter 2B slides.